Bismillahirrahmanirrahim. Today we are going to talk about the cerebellum. Being a part of CNS, it lies inferior to the brain, posterior to the brainstem. It acts as a coordinating center to deal with your voluntary movements. So it is concerned with your agonist and antagonist muscles. So receiving various information through the vestibular nerve and coordinates with the sense of sight, and it has to deal with the tectocerebellar tract by your sight, right? And all these are supposed to make your movement smoother. And as we know that if it is not functioning properly, we will not be able to perform our movements in a fine and smooth way, right? And if it doesn't function properly, we expect there could be hypotonia, some alteration in your gait, and we expect there could be ataxia, nystagmus, and so many abnormalities we can find in our body. And we should know that the lesion of one cerebellar hemisphere is supposed to give signs and symptoms limited to the same side of the body. That is very important for you to learn, because it has to coordinate all reflexes and voluntary muscular activity, right? And through this, it maintains and influence your muscle tone and normal body posture, right? In the picture, you can see that there are two cerebellar hemispheres and a central vermis, right? And there are so many transversely running fissures that divides it into various lobules, right? And we expect a thin cortex that is on its outer side and forming the gray matter, and buried inside centrally placed white matter. And of course, the smaller masses of gray matter we expect as a nuclei are buried in the white matter. And what are they? Can you revise the mnemonic? Doctors eat good food. Yeah, the dentate, the globose, and you expect the emboliform and the fastigial nucleus. These are the four nuclei, a part of intracerebellar nuclei of your cerebellum, right? Yeah, this is the cut section of the cerebellum. Here what you see, the small amount of the white matter in the vermis that closely resembles to that of a tree, showing the trunk and the branches. So we name it as arbor-vitae pattern. And you expect large amount of white matter in your each cerebellar hemispheres, right? This histological picture, you can see that the cerebellar cortex is thrown into various folds called folia, and on its outer side you can visualize the gray matter being divided into three layers: outer molecular, middle Purkinje cell layer, and the inner granular cell layer. And the center core of this folia is being filled with the white matter, right? Yeah, we are looking into the cut section of the cerebellar cortex. The pink color of the core area is showing the white matter, and on its outer side we expect the seat of intelligence, the gray matter. It has got three layers. Yeah, the outermost, which is shown in the sky blue color, is the molecular layer, right? And next to it, or the middle layer being shown with the green color, is called as Purkinje cell layer. Then you see the innermost layer drawn in purple color is the granular cell layer, right? Now we will study the various neurons and their branching processes and the layers in detail, right? Oh, what you are looking? Yeah, you are looking into a sky with so many stars. You are right. This is the molecular layer of the cerebellum, the outermost layer. It resembles with that molecular layer. That is why I am showing you the picture. We know the molecular layer is a synaptic zone, so we expect that so many dendritic arborization of various neurons along with the unmyelinated axons should be running here parallelly to the surface, right? And when these fibers are cut, they appears like a dot, hence molecule. So the name of this layer is molecular layer. Now correlate it with the actual layer, right? Yeah, you are looking into a slide schematically drawn. Yeah, this is the molecular layer, just resembles to that of the starry sky, right? But what it shows very little amount of the cells inside, and the dotted appearance, as I've told, due to the cutted appearance of various fibers. So the cells we have to discuss here are the stellate cells, right? Can you see the stellate cells? And we can then visualize that there is another variety of the cells in this molecular layer is the basket cells. We will discuss them one by one. Then you can see the second layer, which I will show you, is in the middle, Purkinje cell layer, and they are just looking like the beaded or pearly appearance of a necklace, right? Yeah, this picture resembles to that of the middle layer, which is made up of the cell bodies of large multipolar neurons called Purkinje cells, and they form a single row, you would say, along with the outer margin of the inner layer of the cerebellum. So keep in mind this picture, right? Now we come across again with the transfer section and focus on that outer molecular layer, and the layer is being marked with the top word, alphabetic word M, and you are in the molecular layer, which is wider one and showing more processes in it. And the yellow line is showing the stellate cells, right? And how they appear. These stellate or starry shaped pericardia, they have got short thin dendrites and fine unmyelinated axons. We see the ramification of the dendrites, right? And we see that their axons, they are transversely extending, and they have two synapses with the dendrite of the underlying cell neurons, right? And now after that, focus on that basket cells, which are being marked by the orange arrow. These are another cells of your molecular layer, and they've got a small pericardia or small cell body. They have to give rise so many dendrites. Can you see like that of the branches of a tree, right? They have to ascend upward towards the superficial surface, and the unmyelinated axons of them, if you concentrate, they are running horizontally, and they are giving off many descending branches and are forming the basket around the body of the neuron of the second layer, right? Let me mark their basket appearance. This is the basket shape appearance of their descending axon. That is why they have attained their name basket cells, right? You can see that the green arrow with the letter P is towards the central or middle layer called Purkinje cell layer, and here the single row of cells having the cell body or perikaryon. And what type of neurons they are? They are multipolar neurons called Purkinje cells, and their massive flask-shaped cell bodies are arranged in a single row, and they are present in between the molecular layers on its superficial side or outer side, and they are present along with the third granular layer, right? And we expect that these Purkinje cells have vesicular nucleus with the prominent nucleolus, right? And their cytoplasm will address large number of nasal substances and giving it darker stain, and they are arranged around the nucleus concentrically, right? And what else? The dendritic processes, you can see, are arising from the apex of these Purkinje cells, right? And they are forming what? They are forming 2 to 3 large primary dendrites, and I feel that their look, let me mark them. This dendritic process, they appear as a stag horns, isn't it, right? And they are supposed to enter into the molecular layer and branch repeatedly there to form a fan-shaped arborization, right? And their axons, you can see, is arising from the base of the cell and later on acquire the myelin sheath, right? and they have to pass through the underlying granule cell layer and finally enter the white matter. And where they end, the axons of your Purkinje cells have to terminate in the deeper seated cerebellar nuclei. You can recall them dentate, emboliform, globose, and vestigial, right? And the innermost layer, which is again a bit wider, marked with the pink arrow, and it contains a dense population of small neurons called granule cells. And usually in H&E stain, their cytoplasm does not stain well, right? And the granular layer appears darker because of their closely packed nuclei that take deep basophilic stain. And in between them, you can observe the lighter staining area called glomeruli or cerebellar island, right? Now we have to correlate it with some other picture. Now, can you see that these granules or pea-like structure? This is a picture that resembles with the granular layer, the third layer. And in between, the unstained area or lighter stain area are called as glomeruli, or you would say cerebellar islands, right? And in the third or innermost layer called granular cell layer consists of cells, granule cells and the Golgi type 2 cells, right? And the each granule, you would say, cell, they have to give rise 4 to 5 short dendrites that branch within the islands, that cerebellar islands, right? And here they synapse between the dendrites of the granule cell and the fibers coming towards them called as Mossy fibers, which ends up here coming from the spinal cord and the brain stem, right? And the unmyelinated axons of the granule cells, they have to ascend towards the topper layer, the molecular layer, where it bifurcates into two branches. And they are supposed to form the parallel running fibers over there, and hence the name parallel layers, right? And these fibers make synaptic connection with the dendrites present in the molecular layer, and of which cells? The Purkinje cells, right? Another important diagram to make you the clear concept. So look at the blue color mossy fibers. What are they? They basically originate in the spinal cord, so forming the spinocerebellar tracts, or in the brainstem, where they form vestibulocerebellar and pontocerebellar fibers, right? They have to terminate in the glomeruli of your cerebellar cortex, right, which is in the third layer. And at the termination, what happens? The fibers branch repeatedly and give rise to the moss-like appearance. So the name is given to the fibers, the mossy fibers. And we can see the terminal fiber here. You observe they are supposed to make synapses with the dendrites of the granule cells in the glomeruli of the granular layer. And in this way, we have to discuss these mossy fibers. So we can memorize it by the mnemonic. So memorize the line, me at the ground. Me, the letter M for the mossy fibers, at the ground. The word G of the ground is for the granular layer. So the mossy fibers are supposed to terminate in the ground or granular layer. That is why I have told you the mnemonic, right? Now concentrate in the picture for another type of fibers called as climbing fibers. These are the nerve fibers that originates from the inferior olivary nucleus of your medulla oblongata. And when they enter the cerebellum, the fibers run in the white matter to reach the gray matter, or in the cerebellar cortex, where they have to synapse with the dendrites of the Purkinje cells in the molecular layer. So how we can learn that these fibers, where they go? So memorize a mnemonic line that climb to the mountain. So climbing fibers are climbing to the molecular layer, hence the word mountain I have used, right? This is why you can easily memorize the name of the fibers and their termination, right? and again concentrate. They're the granular cells when they are giving dendrites and how they are forming the parallel fibers in the topmost layers, right? Now I would like to show you various histological views. These are the folia, and you can see the outermost covering, or meningeal covering. Next to it, you find the lighter stain topper layer, molecular layer. Next to it, the darker stain, let me mark them, that these are the Purkinje cell layer, and then you enter into the granular cell layer. That was the molecular layer, right? And the area, this area is the white matter, right? Through this picture, again, it should be clear where the white matter lies and where is the gray matter, right? Another beautiful picture to show you the innermost granular cell layer showing the granules and the beaded appearance of the Purkinje cell layer and the molecular appearance of the molecular layer in which fibers are predominantly shown, and when they cut they appear as a small stars, right, to give a dotted appearance. So what else we can learn from all that map? That the various fibers, the climbing, the mossy fibers, they are supposed to have excitatory effect on the Purkinje cells. And what about the stellate, basket and Golgi cells? They may have inhibitory functioning like interneurons. So these interneurons have to do the inhibitory function. But in general sense, the consensus is that the Purkinje axons are supposed to exert an inhibitory influence on the buried neuron of the cerebellar. You would say nuclei we discussed earlier, right? And this is how the cerebellum function as a coordinator for the precise movement of various, we would say, with the help of its output to the motor area and input. They all are influencing and doing adjustment, right? And what is that? That your activity of the low motor neuron is also influenced, rather, right? And there could be the firing of alpha and gamma motor neurons, and we expect that your cerebellum has to send back info to your motor cortex, and in this way they inhibit the agonist muscles and are going to stimulate the antagonist muscle. The main purpose for all this is to limit the extent of voluntary movement and to provide the finer and smoother and coordinated movement in your body. Thank you very much.